Welcome to the DNA of Things, where we explore how our potential is realized at the intersection of DNA, environment, and behavior. I'm your host, Dr. Jeremy Koenig, and today we are joined by Dr. Joe Handelsman, a pioneer in metagenomics, former associate director for science at the White House, and director of Wisconsin Institute for Discovery. In this episode, we uncover how the microbiome shapes our health, the future of personalized medicine, and the role of science in policymaking. You'll walk away with a new perspective on microbes, not as enemies, but as the architects of life itself, and just how do we architect this life between science and humanities and politics. This is a stimulating conversation, to say the least. Let's dive in. Thank you so much for joining us today, Dr. Joe Handelsman, on the DNA of Things. If you don't mind, I'll just run through a little bit of your background here to provide the listeners context. So, Dr. Joe Handelsman, a prominent educator, science policy leader, formerly director then, if you've moved at Wisconsin Institute for Discovery.
Like the director.
It's right. You're still the director there. Still holds a position of U.S. research professor and Howard Hughes Medical Institute professor. Well, you earned your Bachelor of Science in agronomy from Cornell University and your PhD in molecular biology at the University of Wisconsin. How I came across Dr. Joe Handelsman back in my early days as a graduate student is because of this term metagenomics. And, that really springboarded, my own curiosity and pursuit of academia. So it's an absolute pleasure to be speaking with you today, Dr. Handelsman. And, your work extends far beyond the bench. Your stint previously as the Associate Director of Science in the White House Office of Science and Technology under Barack Obama is something I'd really love to dive in today. I suppose also given the, the climate of, let's say the political climate, but the innovation climate, where are we with health? I'm sure you have critical insight that folks would love to hear. But more than anything else, as I mentioned at the outset, Dr. Handelsman is, this is really the story of your journey through science as a researcher, as an educator, and as it turns out as a policymaker. So let me just say again, welcome to the DNA of Things.
Well, thanks for having me, Jeremy. It's a delight to be here.
Mm-hmm. Well, I know we don't want to spend too much time in the small world of microbiology, but in metagenomics. But I would love to extend. Let's just. Most selfishly, I'm interested in how you came to metagenomics. And I'll preface this with saying, as you know, I did my PhD with Dr. Ford Doolittle. I worked previously with one of your postdoctoral students, Pat Schloss, leveraging some of his early day softwares that were, really instrumental to understanding the vast diversity of the molecular world. And so, I was literally standing on the shoulders of giants, you among them. And so I'd really love to understand what the kind of bends in the road were to get you to that point of discovery and innovation.
Well, sure. That's an interesting topic. I could talk about it all day. So cut me off when you get bored.
Mm.
So I was always interested in the interaction of microbes with each other and with their hosts. And one of the ways that microbes interact is with small molecules. And that's essentially the currency of communication and life in a community for a microbe. And so for a long time, my lab studied small molecules like antibiotics that had a direct effect on the survival and success of other microorganisms on plants. And at that point, we were focused entirely on plant roots. And I was very interested in the communities that these microbes have to live in on plant roots and in soil, because the soil microbiome or microbial community is probably the most complex habitat on Earth. It's probably the most diverse group of organisms generally, not just microorganisms, of any habitat. And so any microorganism that wants to get a good meal from a plant or cause disease or whatever it has evolved to do has to be able to first survive and then flourish in this complex community. And what we knew at that point was that many of the organisms in these communities, and soil is one of the worst habitats for this characteristic, were not able to be cultured in our standard culture media, which is in the 1980s and early 90s. That's what we were using. We would culture organisms on petri dishes, and many of them would come up, of course, and grow, but the majority probably didn't. And the estimates for soil are that about 1% will grow on standard media. So we knew that we were looking at a very small subset of the microorganisms that were there. And so if you're studying a community, what are you really learning of substance if you're looking at 1% of the species that are there and ignoring the presence of all the others? And so as soon as the technology came around to look at the signature sequences for microorganisms, so these are the DNA sequences in the small ribosomal subunit gene that is considered a chronometer of life. It basically is a measure of evolution and relatedness of microorganisms. And once we had the method of PCR, polymerase chain reaction, we could go into soil, extract DNA without ever culturing organisms, and then use PCR to amplify all the 16S genes. And that gave us a much more complete portrait of the community and the soil. So we would sequence all of the 16S genes, get a picture of what organisms were there and how they're related, and we could look at change in the composition of a community. But the thing that never gave us was a whole lot more than just the signature sequence. We could say that, yes, this particular sequence and the organism it came from were present. But if we didn't know much about the organism, if it wasn't able to be cultured, then we couldn't say a whole lot about it. And so that was a frustration. Okay, great. It's wonderful. It was an enormous step to have the profile of this community, but we wanted to go deeper. At the same time, my mother had very serious lung infection for several years, and the bacteria in her lungs kept developing antibiotic resistance. And she would be on an antibiotic, and then it would stop working because of resistance, and they'd put her on a different one, and then that one would stop working. And as she was getting sicker and sicker, and my lab continued to work on antibiotic discovery in soil organisms, there was just this enormous irony and frustration that here was my mother dying because of the lack of an effective antibiotic, and my lab discovers antibiotics, but we couldn't find one fast enough. And so a colleague and I were talking about what if we could access all of the genetic potential of the soil? We know that probably 99% of the organisms aren't culturable, but there's no reason to think that the ones we can't culture wouldn't have just as many antibiotics as the ones we can culture. And soil has been the source of most of the antibiotics we use in medicine today. And so metagenomics was the answer to that if we could clone big pieces of DNA into a culturable organism, so again, extracting DNA very gently this time from bulk soil, so we were getting all of the different bacterial genomes present, and then put those fragments of those genomes into a culturable organism, then, if the genes were expressed, then we might see that organism producing a new antibiotic. And that worked to some degree. We did find a couple of new molecules, and a lot of, actually, interestingly, the most, I think, productive part was looking at resistance genes, antibiotic resistance genes. And we did find a lot out about the antibiotic resistance genes in the uncultured community. But again, no method works perfectly, and a lot of the genes that we were interested in didn't get expressed in the cultured organisms. And so we never saw the products or antibiotics. So it wasn't all that effective for what I originally wanted to do to save my mother. And eventually she died and didn't have the antibiotics she needed. But what it did do is that many people were using this method to sequence DNA from the soil directly, and not just sequence the 16S gene, which was the signature sequence, but sequence entire genomes. And that really became the kind of hallmark of what we then called metagenomics, looking at genomics at a meta level, pooling all of these genomes together and looking at them in a totality. And so metagenomics has yielded enormous information about microbiomes in every environment because we can interpret a lot of the genomic information and sequence it even if we can't grow the organisms.
Amazing. Yeah, as you were describing those approaches, it took me back, as I say, to my graduate school days. One of the studies that I was a part of was looking at I would say this wasn't quite, there were a couple of levels of metagenomics, let's say, and I don't want to use the term incorrectly. So there were instances where I would, extract full DNA from, ocean samples or, let's say soil samples. And I was specifically interested in a region of DNA, a hypermobile genetic element called the integron. And so, essentially what I would do is I would take those constructs of, DNA and then put them into, a host organism and then screen the host organisms to see if they contain the integron. And one study that we did was in the barrier reef. We were actually looking at the coral microbiome, if you will. So we were actually extracted coral mucus out near Townsville, Australia. And as I say, from there, we isolated metagenomic samples and then, screened isolates for the conserved region of the integron gene. But then we would sequence these whole, phosmid, big libraries of DNA. And it was clear that it was what was prevalent in those coral microbiomes, at least the organisms that had the integrons, were these some pathogenic, some non-pathogenic Vibrio species. So Vibrio fulmificus and Vibrio parahemolyticus, for example. And we would look at the integron gene cassette. So there's an array of genes, some of them, 200 genes long or more. And it was interesting, like you mentioned, those housekeeping genes like 16S, and, we would, cluster them according to those, let's say, conserved taxonomic markers. But then what we would do is we would classify the relatedness of the integron gene cassettes. And what we were effectively doing was demonstrating how dynamic that region of the genome was. And actually what we were seeing, like to your point, we would see antibiotic resistance markers, but, the microbial, what would you say is biological warfare is going on there, right? Because all kinds of these antibiotics are actually being made by these other organisms to kind of, jockey for their niche in the microbiome or, actually invade the environment. But what was interesting is when we compared the coral pathogens or the benign organisms to human pathogens like cholera, Vibrio cholera, for example, we actually saw identical gene cassettes in the cholera, demonstrating, is this a gene highway for antibiotic resistance, and really just pointing to the fact how, ingenious nature is. And so to have those tools and to go in and to tell a story there and to get a better understanding of how accelerated evolution can happen, especially in the presence of antibiotics, it really gives you some kind of idea into how you might actually use that information to make better informed protocols / policy around how do we use antibiotics, right? Like, don't oversubscribe them. Do we subscribe them or prescribe them in a particular time course? Geographically, do we have a coordinated plan in terms of how we use antibiotics globally? That's where my head always went, especially when I went into the world of the gut microbiome, work that I did with Ruth Lay. And we looked at how the infant gut was colonized over 2.5 years. And so when you were talking about just the dynamic nature of the soil, like something came to mind. There's actually, two questions that are kind of burning here. And So the first one is maybe more of an industry question, and then the second one is more of a policy question. And we can maybe jump from one to the other. You pick which one first. But on the side of applying metagenomics, let's say in the commercial setting, you see these different companies popping up offering you your gut microbiome snapshot. How useful is that versus entertaining? And then maybe, springboarding into, science and policy, because I suppose there's a lot of, interfacing not with just the academic world, but the industry world. And, there's not many people I get to speak to who can speak on both levels. So feel free to grab whichever one of those questions makes sense.
Well, in terms of getting microbiomes sequenced, this is a really interesting question that gets asked all the time.
Cool.
And I'm probably a little bit conservative about it. Other people, other scientists are much more willing to interpret the meaning of the microbiome in the human gut. I would say what we can safely say is that high diversity seems to be associated with healthy microbiomes, and lack of health seems to be associated with a diminishing diversity or fewer species. And so how you get that diversity is usually from a diversity of fiber sources, because fiber is what bacteria eat. By definition, fiber is something we can't digest, but it gets to the gut and the bacteria can. So that's their food. And there are many different kinds of fibers in plant material. And the more different kinds a person eats, the more species rich or diverse their microbiome. So that's something I think is pretty safe to conclude based on current data. There are also certain patterns that seem strongly associated with certain diseases, like Parkinson's disease and many other obesity and, a whole diversity of diseases. It's not just one class of disease. And so we know something about those patterns, but we can't at this point use most of them for either diagnostic purposes or remedial processes. There are a few papers, for example, with Parkinson's showing the use of probiotics to modify the gut microbiome. And I've heard that some of those are very successful, even beyond the papers, which show pretty good effectiveness, that they reduce the symptoms of Parkinson's. So that's one where I think we have a pretty good association. But just getting any average person's microbiome sequenced is partly useful, but not, in my mind, that interpretable yet. One example I heard was someone who had the metabolome of their microbiome done, so looking at all the metabolites, and they found no butyric acid in the metabolome. And that is one of the most essential small fatty acid in the human body, because the only food that the gut lining cells, the epithelial cells that line the gut, will eat is butyrate. And most of it comes from the bacteria in the gut. So if this person didn't have butyrate and clearly had a microbiome that wasn't very good at producing it, that was a problem. And that is an interpretable one, because that's a metabolite that is extremely well studied. And we absolutely know that it is necessary for the health of the gut. And even beyond just the epithelial lining, there are associations of all these different diseases with a lack of butyrate producers in the gut, including brain diseases. And it looks like butyrate is important probably for keeping the gut lining healthy so that certain things don't escape the gut and end up in the brain, usually chemicals. So that's something that is under very active study, something my group is studying now with respect to depression, looking at the association of the gut microbiome composition and depression and responsiveness to various treatments like antidepressants or meditation. And we found, for example, that meditation changes the gut microbiome. Wow. So I think we know a lot about the microbiome, but I think the average person getting their microbiome done gets interesting information, but maybe not infinitely interpretable unless they have a particular ailment.
Yeah, you mentioned the correlation of different communities of bugs with the profile of short chain fatty acids. I mean, that was something that we certainly saw as like positive and negative correlations. I don't know what the predictive power of that is. Again, I think of like this study that we did was really more, as a perspectival, as to kind of take a step back and, see it as more hypothesis outcome versus going in and testing a specific hypotheses. But, we started to see observations like, okay, well, as you say, from birth, the diversity is very small in comparison to the mother, very low. And you see the general trend up, but then there's like perturbations throughout, right? So switching or introducing, for example, solid foods, antibiotics, introduction of cow's milk, just, again, just measuring simply the change of fluctuation in diversity was essentially consistent with what you're saying. And then actually going in and looking at the functional gene profiles to get us understanding of what's going on there at the biochemistry potentially for the metabolome. And, it's interesting because there are so many of these different studies that I remember back in the early days of this, where, the correlation of one specific microbiome with obesity and one with lean, and doing these transplant experiments and bringing about changes in phenotypes. So it's like, hey, if I give you a lean gut microbiome, at least in rats, or seeing the reduction in obesity. I think there are experiments also where they would stress out mice, like get them floating on sponges and then take the changed microbiomes and put them in other mice and then cause a stress phenotype, which is pretty amazing. And, it immediately gets me, asking the question is like, it's a kind of a chicken or an egg type of thing, right? In one instance, your environment caused this change in your gut microbiome, but then in the other instance, you get exposed to a microbiome and it causes a change in your phenotype. So, I mean, one thing that I have seen in terms of like actual, let's say, clinical application of this is in cesarean births, right? So by bypassing the birth canal, you don't get exposure to the natural gut flora from the mother. And then that has a increased incidence of, asthma and chronic inflammatory diseases. And so implementing these kind of like vaginal swabs for the babies who are born by cesarean. Are there any other like clinical applications that you're seeing now? I know one that they talked about during, when was it, the Rio Olympics, right? Was, athletes were getting the flu. I heard like, some very kind of biohacking athletes had like a stored healthy sample of their gut microbiomes that they might take with them for performance purposes. But yeah, are you seeing any like clinical application of the consortium rather than probiotics or drugs?
Well, one interesting discovery that I think will eventually lead to manipulation is that if you take, and this answers your question about sort of chicken and egg, which comes first, what's causal. If you take the microbiome from depressed people and put that microbiome into rats, the rats will become depressed. And to me, that's pretty powerful evidence that the microbiome is not just a result of the depression, but it is actually causal to it. And I think that's an area, if that's true, that will be the basis for all sorts of manipulation in the future. But some things are already there. I mean, so one, I think, pretty standard use of large-scale manipulation is fecal matter transplants, which are exactly what they sound like. And those were first used for Clostridium difficile, or C. diff, which is a horrible gut pathogen that establishes when people have been on long-term antibiotic therapy. And it tends to reduce their normal microbiome. And then this C. diff, which is present in many people's guts, but it's just kept at low levels when there isn't a strong, diverse community to keep it in check, will start taking over, and it causes extremely severe symptoms and eventually death. So some very brave scientists took some patients who were literally 24 to 48 hours from death from C. diff, and they got permission to do fecal matter transplants. And these people who were literally on death's door got up and walked out of the hospital within hours. It's one of the most remarkable treatments I have ever seen. Most fecal matter transplants have not been quite as dramatic, but one study that I thought was very good was on autism, where they did fecal matter transplants to very young children. I think they're about 2 years old with autism, and they used fecal matter from healthy kids that were not neurodivergent, that had normal, so-called normal behavior. And they found that after a year, the kids who had gotten the transplants had many fewer GI symptoms than the cohort that didn't receive fecal matter transplants, which is the GI symptoms are a typical side effect of autism. And then after another year, there were significant and it seemed like pretty substantial changes in behavior rated by an outside person who didn't know what treatment they had gotten, rated by parents, by teachers. And so that suggested that the behavior was being influenced, at least in part, by the gut microbiome. And I found that to be pretty remarkable. I think anytime the gut microbiome influences behavior, clearly interacting with the brain, I just find that a fascinating connection. So there are several like that, where people are trying to do very whole scale level, very large level replacements of the microbiome. But those have not been approved for clinical use for very many diseases. I think C. diff is probably still the major application that's approved by the FDA.
Mm. When you describe that, observation, it makes me think of one of my favorite science writers is Stephen Jay Gould, and he talks about like this notion of progress, right? Are we the most advanced species at the top of the chain? And, because microbes are, so small and so simple. And it's like, well, first of all, let's remember we've been here around the same time and, invoking different evolutionary strategies. And it's like, you kind of tell that story. And it's like, wow. So, really a bunch of microbes can really just get together and, colonize a human. And it's like, here we are now, these walking incubators.
Remember, they were here billions of years before we were. So they,
right.
you could say, are far more highly evolved. We like
Right.
to think we're the most evolved species, but they are far more evolved, and their relationships with each other and the formation of communities of microbes is very sophisticated and very far advanced. So yeah, we like to think we control our own behavior, and our health is controlled by our lifestyle. And in fact, I think a lot of it is microbial. I teach a course in forensics to law students, and I always tell them that someday you're going to see the replacement of the Twinkie defense. If you remember that from many years ago, people are going to start saying, my microbes made me do it.
Right.
We have evidence that microbes affect behavior, and including aggression, at least in animals. So so blame your microbes for anything you don't like, but give them credit for the good stuff as well.
Right. It goes both ways.
Yeah.
Okay. So that's super, far out and far reaching and, just mind opening and altering. And so somebody with your perspective and somebody with, your appreciation of these, complex systems, now you find yourself in the White House in science policy and, working as the associate director for science in the White House Office for Science and Technology Policy. And what is that like? How does, so I'll preface it by saying this, like when I look at, I apply for a grant and then I do the research and then the work is done. And, I think as an academic, we can feel quite, satisfied with a publication that's well received by our peers. But, given it's in a lot of instances public money. It's like, well, where is the knowledge translation, and how is this making people better, right? So that's kind of one question that I've always been interested in, as a former academic, but then just purely curiosity, like what is it like being around those people like who think differently, who I think are more in kind of an applied mindset versus a basic researcher paradigm. And yeah, anything that you can share about that would be very interesting to listeners, to say the least.
Well, the Office of Science and Technology Policy at that time was run by John Holdren, who is a very, very data-bound scientist, the best of the best. He always wanted to see the evidence and be absolutely sure of anything he was advising the president about. And now, today that just seems so incredibly refreshing, because we're seeing so much movement away from facts and data and respect for scientific evidence. But that was the mood in the office as a totality. Many of the people who worked there were scientists. Most of them had some kind of science background. Some had moved into policy and did that exclusively after their training as scientists. Some did it more mid-career. Some did it more temporarily, like I did. I was there 3 years, and then I went back to academia. And so there was a whole mixture of people at, various levels of embeddedness in policy and thinking like policymakers. But what I found, at least for myself, that was the most challenging was that all policy is a compromise. Everything that is done in lawmaking or, creating statements for the public is going to be a compromise, because it's not going to be tied entirely 100% to the data. There have to be political decisions made, and it has to have elements of, well, why are we doing this? What's the benefit for people, Americans, or across the world? And so there are obviously things that are balanced and weighed. And so that can be very frustrating as a scientist, where you're absolutely data bound and all policies in your mind should be based 100% on the data. But sometimes the White House will take a position that has other elements besides the data driving it. And that's political reality. And other people made those decisions. I, for the most part, didn't have to. But that was the hard part. The exciting part was taking science to a level where it really affected people's lives, and then taking policy, inserting policy in a positive way into the lives of researchers, and in some cases making it easier for them to do their work with less bureaucracy, less time spent on administrative tasks. We got rid of a few things that weren't very useful to relieve the burden of a lot of paperwork on researchers, which the burden is enormous. There was an estimate recently that most faculty spend 40% of their research time doing administration. Mm-hmm. And that seems to me like a not very healthy trade-off. It seems we want our scientists doing more thinking about science than administration. And so that was something that John Holdren was very committed to, was making the scientific enterprise work better. And so, and then of course, a big part of it was interpreting science for the president and explaining, what is genomics? How is it going to be used in human health? What should we be worried about in the future in use of genomics in terms of people's privacy and data security? And there were all sorts of issues, which frankly most of them are still alive today, even though it's 8 years since I was in the White House. Many of those issues persist and have actually become much more intensified as data has just exploded in terms of the amount of data about people that is available in the health sphere. So, and then, one of the things that was very satisfying to be part of, I was in no way responsible for it, but the United States response to the Ebola epidemic, our job was Liberia. So we worked in Liberia, and then France took Sierra Leone. The different countries had different responsibilities for the countries where the infections were occurring. I was just so proud of the United States and the United States government and the response to that epidemic. It was, thousands of miles away from most Americans, and most, couldn't even fathom having a virus as horrible as Ebola being spread through our population. But we took it quite seriously, as if it was our very own. And that's what President Obama wanted. He wanted us to treat it as if it was our own infectious disease, because he thought it was so important to use both American expertise in managing epidemics to help other countries, but also to prevent epidemics from spreading further. And so it was awesome to watch the response of the first meeting I called was of all the agencies interested in Ebola that had some stake in it. 26 different government agencies showed up at the meeting. And so essentially every branch of the military, NIH, the CDC, USDA, I mean, you go down the list it was a who's who of the US government. And I was just astounded that all of them felt that they had a stake in this and could contribute, and they did. And within about a year of our engaging with Ebola, Liberia was declared Ebola free. And so, I didn't make the policy. I just facilitated bringing the right people together, asking questions, writing memos for the president. But even though I was just part of it in a teeny weeny little way, it was an amazing thing to watch. And I will always treasure that, because it's such a great example of what United States science and ingenuity can do to help people in other parts of the world. The original prediction was that there would be about 100,000 deaths if we left it alone. In the end, there were 11,000, which I don't think is a great thing, but it was a whole lot better than it would have been if we had engaged. So I was proud of us.
So would it have been hard for you to be on the sideline for COVID?
It was challenging. We left a lot of materials from the previous administration for future epidemics. And my sense was, it was hard, being 1,000 miles away from Washington and not knowing what was happening inside the policymaking rooms. But it seemed like that information wasn't being used. And so that was kind of frustrating, because we did try to make Ebola and then Zika kind of the playbooks for future administrations and future epidemics. So that was hard. It was very hard to watch the change in how the public treated Tony Fauci, who I absolutely revere, loved working with him when I was in the White House. He's one of the most amazing intellects, communicators, and just people that I've known in science. And he seemed to go from hero to villain almost overnight, in a way that I never understood. One day he was like, his face was being put on the tops of cupcakes because he was such an American hero. And then somehow something turned, and now, he has to have security around him at all times because he and his family have been threatened repeatedly. And I just find that very tragic. And I still don't quite understand why that happened. He probably made a couple of mistakes in policy, but that's what happens when you have incomplete information, which with a new virus, of course, was the case. But he saved so many lives. It was awesome to watch him in action.
I look at one of your, passions that is, self-evident, and that is for, education. And that kind of comes through here too. Again, we were talking about like knowledge translation, and there's, it's certainly education of the current administration, but also there's, education of, the next generation. And, I'm particularly interested in that as, I now have a a young daughter who's 16 months today. And, I'm constantly, thinking about, the balance of, exposing her, to the right content at the right time, providing a constructive environment where she can experiment and, discover her own unique abilities, but, aimed at a higher good. And, gosh, I look at the time and we're already at the top of the hour, and I knew this would be one of those kind of 3-hour potential conversations. So I am going to certainly follow up, but I would love to get a sense of what you're thinking in terms of intergenerational knowledge transmission, if you will, in terms of everything that you've learned, how people like, again, current administration, the next generation can really kind of, upgrade based on all the learnings from this generation. And, how do we get there faster, so to speak? How do we get to the truth, faster? And, is there a place for, obviously AI in this? We didn't even touch on that, but, really, if I could get you to, kind of close out with, your vision for education, because, you've got this Tiny Earth initiative, you've got, gender bias in science. These are, like I say, topics each that could be 3 hours, but I'd love to hear a little bit more about the dedication that you put aside for education.
Well, I think in terms of the intergenerational issue, the most important thing that educators and parents and all citizens can do is foster creativity and curiosity, in particular, in our children. And that's the first part, is making them curious, because I think every kid is a scientist naturally. Mm-hmm. And then school tends to take their excitement away from them rather than encouraging them. And not always, of course. Some are absolutely inspired by school, but many are not. The other thing that at the later end that I think is critical, not your daughter's age, but later on, is teaching students of all ages that failure is part of science. Because we see a very strong gender split and racial split and all sorts of different kinds of backgrounds. So, for example, children of scientists are far more likely to make it in science, keep going in science, whereas people who come from non-science families are much more likely to turn to other fields. And even though they may have the same grades, the same performance, they think that, I think one of the reasons, and this is a hypothesis to test, I think one of the reasons is that failure is such a natural part of science. It's probably, more than 95% of what we do. Experiments fail and grants don't get funded and papers don't get accepted and ideas are wrong. I mean, if we knew all the answers, we wouldn't do experiments, right? Mm-hmm. We would just say what the answer is. And if that's not evident to students who haven't grown up among scientists, and they think that every failure is a reflection on their character, their abilities. And so I think that a lot of students drop out of science, even though they're really good at it, because they just don't have a confidence in their own abilities. And we need to correct that. We need to make sure that they don't think that every scientist who has made one of those great contributions that we hear about in high school and college classes got there the first time around, or that they never had a failure, or that it wasn't incredibly hard work to achieve what they achieved. So I think fostering curiosity in children and then teaching them about the role of failure in science are probably the two most important things we can do to create the next generation of scientists.
Oh, incredible. I do have, one bonus question that I forgot to bring up, because you talk about, well, I love what you're saying. It's like, I'll put it another way. It's called research because we have to research. Yeah. And so, on that point, I think about the Human Genome Project, like when it was the first deliverable, the human genome being sequenced, right? But now here we are 20 plus years later, and we've got this All of Us project. right? And I think you were instrumental in getting that off the ground, and maybe that's a great way to kind of segue out is what are your hopes for that project? Insofar as I think it's it's looking at 10 million people instead of one person and a diversity of folks. How do you see that contributing to the understanding of the human condition?
Well, I think it will generate many associations between our genomes and our ultimate conditions. One of the really early discoveries, which it's a funny one, and not all that useful, but it was interesting, was that they found a genetic marker that seems to be associated with liking or disliking cilantro. And that's the kind of thing, who would have thought that would come out of the All of Us project? But of course, there are much more serious correlations that are beginning to emerge about interactions between lifestyle, genes, and disease or avoidance of disease. And so I think there's a lot that will be generated that will be very useful for exploring those connections further, and then ultimately making recommendations to people. Where I see the million-person analysis falling down is that the only omic, so to speak, that they're doing is the genome. And that's partly the time that it was started and who started it and all that kind of thing. I mean, there are lots of reasons for that. But we now know that the microbiome, which is not the genome, it's the microbes' genomes, and the metabolome and the epigenome and all these other kinds of omics have as much predictive capacity, in fact, in some cases, much more predictive capacity about disease conditions and health conditions than the human genome itself. I mean, when you think of it, the microbes in the gut contain probably, I don't know, I think somebody calculated 100,000 times more genes than the human genome has. Mm-hmm. And so, of course, there's more variety and more genetic capacity there. And so that's where I see the next big step is going really deep into the other omics, as well as using things like wearable technology to measure ongoing characteristics and features of human health. Bringing all of those together and then using AI and machine learning to analyze the vast amounts of data that will come from those studies. That's what I see as the next big step. And all of us will provide a nice, in a way, if I can use the word, I don't mean it disparagingly at all, a superficial view of human characteristics associated with health and disease. And then we probably need a much smaller cohort, instead of 1 million, perhaps 10,000 people, to dig really deeply into other characteristics. And I think that's going to yield just amazing insights and ultimately diagnostics and prevention strategies and treatment strategies.
Mm-hmm. Yeah, really the age of, proactive health is upon us, right? It's like, compare that with, I should say, pair that with AI, pair that with your family health history, pair that with your GP visits, and the real-time data, the temporal analysis is like, there's a community of selves that you can compare yourself with, and then, to other more similar relevant populations. So as I said, Dr. Handelsman, we could go on forever. One final question I have, I asked this of all my guests, is who would you nominate to be on the DNA of Things podcast?
I would nominate first Alan Alda, who is usually on the other side of these podcasts because he has his own podcast and asks the questions. But I think he is an amazing scientist as well as interviewer. And it'd be fascinating to hear what experiences in science are the most important to him or which ones transformed him the most. And another person I would pick would be Nancy Hopkins, who is a very great geneticist, worked on cancer and a zebrafish model for many years, but has also been an absolute transformer of the role of women in science. And she's incredibly eloquent and wonderful to listen to. And if you don't have time to interview her, then you should read the book about her experiences as a woman in science called The Exceptions.
Wonderful. Well, I'll make it my aim to do both, to invite her and read the book. I think it's so important to, let the, general public get a better understanding of what goes on in the bench and, well, what goes on in the White House, as it turns out, and what goes on in the field. Because, there's so much to appreciate when you think about when it comes from, idea to discovery, and then implementation. How does this make human life better? So, Dr. Jill Handelsman, I want to thank you for that, for all you've done to make human life better, and for spending an hour of your time with me today. I can't thank you enough.
Well, thank you. Those are great questions and a really insightful hour. So thanks a lot for having me.
All right. Well, that's it for today's journey into the DNA of things. We've covered how microbes shape our health, how science influences policy, and why embracing the complexity of the microbiome could revolutionize medicine. Today made you rethink the unseen forces shaping your life. Please share it with a friend to discuss and to keep the conversation going, because the more we understand the invisible world within us, the closer we get to unlocking our full potential. So thanks so much for listening, and feel free to check out our Substack on the DNA of things. In the meantime, stay curious, and we'll see you again soon.